Welcome to the Jackson Laboratories Humanized Mice PubCast, where we offer a glimpse into the world of humanized mice through the window of a recent scholarly publication. In this series, we're defining humanized mice as immunodeficient mice that have been engrafted with human CD34 positive hematopoietic stem cells or a human PBMC-derived humanized immune system. JAX is a longtime leading provider of both naive immunodeficient and humanized mice to the life sciences community worldwide. We regularly run preclinical studies for external clients using these models, most commonly in the fields of oncology, immuno-oncology, and transplantation biology and drug development. But we're always excited to see the new and different ways that other researchers use these models. Join us as we explore the cutting-edge research and applications of humanized mice.
Hello, everyone. Today, we are going to look at an article which was published in Nature Communications in 2021 by researchers from various institutions within the US as well as Austria and Germany, with a mix of authors from both biopharmaceutical and academic organizations. They created a humanized melanoma model using NSG mice from the Jackson Laboratory to investigate the role of mast cells in anti-PD-1 therapy resistance. Before we look into the results of this study, let me introduce immunotherapy drugs called immune checkpoint inhibitors. These drugs act by blocking the binding of immune checkpoint proteins present in T cells with their partner proteins present in the cancer cells. Since the immune checkpoint protein does not bind to its partner protein, the T cells are not switched off, hence they have the ability to attack the cancer cells. One such drug is the anti-programmed cell death protein 1, which is also known as anti-PD-1. This drug prevents the binding of PD-1 protein present in the T cell with its partner protein PD-L1, which is present in the cancer cell, thus allowing the T cells to attack the cancer cells. Anti-PD-1, with a high response rate and very low side effects, has been approved by FDA to treat advanced melanoma. However, immune non-responsiveness and therapy resistance are two stumbling blocks in the treatment of patients receiving anti-PD-1 therapy. Tumor-infiltrating immune and non-immune cells are known to modulate therapy responses in many solid tumors, including melanoma. Mast cells are granulocytes derived from CD34 expressing bone marrow myeloid precursors, which circulate in the bloodstream and migrate to peripheral tissue and get matured there under the influence of chemokines and cytokines. Mast cells directly impact tumor cells as well as immune and non-immune components of the tumor microenvironment through chemokine secretion and release of other mediating factors, leading to varied cancer-promoting or cancer-suppressive properties. Hence, the authors hypothesized that deletion of mast cells from the tumor microenvironment may improve the efficacy of anti-PD-1 therapy. In order to address this hypothesis, the authors used extremely immunodeficient mice known as the NSG mice to create this melanoma model. These mice carry two mutations on the NOD genetic background. One is the severe combined immune deficiency mutation, which renders these mice T cell and B cell deficient, and the other is a complete null allele of the IL-2 receptor common gamma chain. Now let us look at the various steps involved in the humanization process. 6 to 8 weeks old NSG mice were myeloablated using busulfan at a dosage of 30 mg per kg body weight. After 24 hours, these mice received a thymus graft in subrenal capsule, which is capable of producing thymic organoids, which would support human T cell development. This is immediately followed by injection of autologous liver-derived CD34 expressing hematopoietic stem cells to accelerate human immune cell reconstitution. Between days 6 and 12, these mice received AAV vectors, which delivered various interleukins and stimulating factors, and DNA plasmids, which delivered stem cell factor, thrombopoietin, and FLT3 ligand. At day 50, increased levels of human immune cells from various lineages were observed in this model using multicolor flow cytometry. All reconstituted mice were assigned into experimental group based on the levels of human immune cells. The mice were subcutaneously injected with HLA-A allele melanoma cells over the right flank, as shown in figure 2, and the growth was monitored. After the tumors became palpable, starting from day 7, the mice were treated with 5 to 6 injections of intraperitoneal anti-PD-1 with a dosage of 10 mg per kg body weight for a week, as shown in figure 3. And the respective IgG antibody was used as a control at similar dosage and frequency. Now let us look at the results of the study. The authors used a digital caliper to measure tumor growth. Humanized mice with T cell reconstitution, as you can see in this image, it is denoted by a blue line. Before anti-PD-1 injection showed significant tumor growth restriction when compared to non-reconstituted NSG mice, denoted in this image by brown line, and humanized mice with high circulating B cells, denoted in this image by magenta line. Tumor growth was then measured after anti-PD-1 therapy. The two graphs on the screen denotes tumor growth data from two different melanoma tumors after anti-PD-1 therapy. As you can see, anti-PD-1 therapy restricted the tumor growth of both the melanoma tumors significantly, which is denoted by blue line, when compared to humanized mice treated with control IgG, denoted by magenta line, and non-reconstituted age and sex matched NSG mice treated with anti-PD-1 therapy, which is denoted by brown line. Now that the authors have demonstrated there is tumor growth restriction after anti-PD-1 therapy, they went on to look at the immune phenotype present within the tumor. To determine the immune phenotypes within the tumor cells, immune histology staining was performed. The presence of mast cells was confirmed using mast cell tryptase staining. The two images on the left side shows sections from untreated tumor, while the two sections on the right side shows sections from tumors treated with anti-PD-1 therapy. As you can see in this image, the untreated sections show negligible staining for mast cells, while the treated sections shows robust staining for mast cells. If mast cells are associated with anti-PD-1 therapy resistance, then their depletion should alter tumor growth. Mast cells are known to be c-KIT receptor positive, and one can target these cells by pharmacological intervention using small molecule inhibitors. Hence, the authors use sunitinib, a multi-targeted receptor tyrosine kinase inhibitor, and imatinib, which is similar to sunitinib, but it is more specific to c-KIT receptor. Inclusion of sunitinib and imatinib along with anti-PD-1 therapy resulted in complete regression of tumor in mice, and treatment only with sunitinib, imatinib, anti-PD-1 and control IgG did not influence tumor growth as seen in figures A, B, and C. Meanwhile, cediranib, a drug targeting VEGF and PDGF receptors, failed to cause complete regression of tumors when included with anti-PD-1 therapy as seen in figure D. This again proves that mast cell depletion causes tumor regression. Sunitinib treatment along with anti-PD-1 therapy also improves survival rate as evidenced by the survival curve. Survival curve shows survival advantage in tumor-bearing humanized mice that were treated with sunitinib in combination with anti-PD-1 therapy, denoted by black line in this image, when compared to mice treated only with sunitinib or only with anti-PD-1 or only with control IgG. Thus, the authors conclude with the note that using c-KIT inhibitors such as sunitinib improves the efficacy of anti-PD-1 therapy by depleting the mast cells, resulting in regression of tumors and increase in survival rate. The authors have also studied the mechanism through which mast cells cause this therapy resistance and demonstrated mast cell infiltration in human tumor samples as well. I suggest you all to refer to the manuscript for more details. I would like to re-emphasize the benefits of using a humanized cancer model. The authors were able to mimic the human tumor microenvironment to obtain reliable and translational in vivo data using this humanized melanoma model. Co-transplantation of human fetal thymus and liver-derived CD34 hematopoietic stem cells led to the development of sustained human hematopoiesis and a functional human immune system. This helped the authors to properly investigate immune-based therapeutics for cancer treatment. This article was published in the year 2021, while in 2022, we added two new NSG strains to our repository. One is the NSG SGM3 IL-15 strain with the strain number 033216. This strain expresses human interleukin 3, interleukin 15, granulocyte, macrophage, colony stimulating factor, and stem cell factor. This strain combined the features of a highly immunodeficient NSG mouse with human cytokines that support the stable engraftment of myeloid lineages, regulatory T cells, and NK cells. The other strain is the NSG FLT3 mice with a strain number of 03337. So this strain has a transgene which expresses human FLT3 ligand and a knockout of its receptor in the NSG background. Using these two strains, researchers can eliminate the extra step of delivering human cytokines and chemokines using vectors and plasmids separately. I noticed that the strain number was wrongly cited in this manuscript. It might be a small typo error. In case if you are interested in using this strain used by this authors, the strain number is 005557. Let's all meet in another episode. Thank you.